welcome back from the breakout rooms, everyone. I hope you had a chance to meet some new people, learn about vacation spots and cities around the world. I certainly learned more about York than I ever imagined I would in that 10 minutes. In terms of our program today, we're going to hear first from our Platform Scientist Award winners before we get to our guest presentations. So at this point, I would like to invite Gareth, who works at the Ottawa Hospital Research Institute, to share his presentation. Gareth, are you there?
I am here. Let me just figure out how to share appropriately. 1 second.
Sure.
There we go. I think I am sharing the screen now.
Okay, I don't see it quite yet.
There it goes. There it goes. Just a moment. Yeah, my machine's slow right now.
No problem.
There we go. Is that visible?
I can see it says Gareth has started screen sharing. Can anyone see the presentation yet? Oh, there it comes.
Yeah, I might. Okay, so that's okay. Great. Okay, well, thank you very much for giving me the opportunity to present. I'd like to take a moment to describe our core. The Ottawa Bioinformatics Core Facility is a core facility of the University of Ottawa and the Ottawa Hospital Research Institute. I'm showing here an image of the Faculty of Medicine on the General Hospital campus, and that's one of the funding groups, but we're actually located in a separate building at the Ottawa Hospital. Most of our clients are Ottawa Hospital Research Institute or the University of Ottawa faculty, but we've had clients from around the world over the years. Dr. Perkins, Dr. Ted Perkins is the scientific director of our core facility. The staff consists of just 2 full-time bioinformaticists, Chris Porter and myself, and I do double duty as the manager of the core facility as well. Both Chris and I have been working at the Ottawa Hospital Research Institute and doing bioinformatics for about 20 years now. We've done quite a range of bioinformatics and other analyses during that time. Chris in particular is very experienced and has been doing bioinformatics since before it was a thing. He was doing it way back in the 90s before it had a name. So we do a variety of things and provide a variety of different kinds of support. Where we really like to start engaging with clients is in grant application support. If we can get in at the grant application level, we can help design experiments. We can help optimize the sort of money that's spent to test hypotheses, doing proof of concept analyses, and really refining how the effort is spent on the analysis part and the data generation. Unfortunately, we don't always get in necessarily at that point. It's not uncommon to have researchers come to us with data that is perhaps not adequate to test the hypothesis that they had hoped, but we do the best we can. Most of the analysis we do, which is the bulk of our work, is working with next generation sequencing of all different types, ChIP-seq, RNA-seq, TAC-seq. There's all kinds of different experiments you can do that read out as sequenced cDNA of various types. And then we do multimodal data integration, statistical analysis, experimental design support, whatever needs to be done. Training has become more and more important for us in terms of the value we provide to the organizations who fund us, particularly over the pandemic. What happened during the pandemic is we had been doing this stem cell research supported 3-day bulk and single cell RNA-seq workshop, which we would do 1 time a year. And during the pandemic, we were asked to do it online. And then because it was far cheaper to do it online than fly everyone in, we were doing 3 instead of 1 a year, and the demand kept up with it. So it's proven to be, well, it's not quite as good as doing it in person. Doing it online is a very effective way to train students. We've also done our training with Stem Cell Network and BioCanRx, both of which are network centers of excellence in Canada. This year, we worked with Dr. Perkins to teach a course at University of Ottawa, and we'll also do individual student postdoc training. Often that'll take the form of working the student or the trainee through the analysis that they're doing. So when they're done the analysis of their data, they fully understand the code associated with it and fully own that analysis. And then the final thing we do a surprising amount of is what I call scientific computing support, applications configuration, support and troubleshooting. Because we're located at a hospital, the IT isn't well equipped to do the kinds of things that are necessary for instrument control systems, for example. So we'll often work with the labs to do that. So my role, I'm a bioinformaticist and a little bit of administration, or because I'm in the role of manager in the Bioinformatics Core. For the bioinformatics, as I said, mostly sequencing analysis of various types, RNA-seq, ChIP-seq, whatever. Scientific administration and scientific computing support is a pretty common one that I'm doing. So yeah, as described just before. So a lot of work keeping systems running, or firewall may break them, for example. So working with the hospital IT to get things working again. We do a lot of training. On the administrative side, as you're all probably familiar with, accounting, billing, reporting, workshops and training organization, and what seems to be an endlessly growing number of committees that I'm expected to be on. One thing that's also been picking up is research data management issues, which are associated with IT in various ways in the bioinformatics. So our challenges, briefly, the challenges we experience in the core, client recruitment and sales. As many of you who work in core facilities are familiar, scientists are very busy. Running a lab is several jobs in one. And while there are many labs we could support, the scientists have a lot more to do than necessarily seek out yet another group that could maybe or maybe not help them. I'm not sure. So engaging new customers is a difficult thing. We found training really helps with that. And I'll talk about that later. System administration, because we're in bioinformatics, we're dealing with a lot of computing and data, and previously used to run our own clusters. Now we do everything on Compute Canada. Moving to Compute Canada, I had hoped that would reduce our system administration, but it seems to have just displaced it to other things in various ways. So there's just a component of keeping the systems running and keeping our pipelines running, etc. As mentioned briefly, our hospital, because we're located in a hospital and the security restrictions of a hospital are rather different than that of a research organization. We have a rather more restrictive environment, and working with that can be difficult. So that's been something we've struggled with. And finally, research data management is something that's picking up, and just dealing with all the data that needs to be dealt with in the various labs, and ensuring that requirements as specified by the Tri-Council are fulfilled is something we're trying to make sure that we're able to do and provide help for. And I'm just trying to change the page here. Okay, so our successes. And finally, I'd just like to talk about our successes. Training. Training has been ramping up for the last years since the pandemic. A lot more online training, which turns out to be very effective. It's also a good way to work with students to do their own analyses. Once again, not as effective as sitting next to someone or sitting in front of them, but still works. We've learned to focus on practical analysis with students. I like to say it's the difference between, in many cases, bioinformatics people will teach the abstractions of it for the purposes of students' analysis they want to do. They're far more interested in just practically getting their analysis done. So we teach the theory, but we really like to focus on getting that analysis finished and how you do that. So it's like the difference between teaching thermodynamics or how to fix your engine. You can teach a little of both, but we really like to emphasize on the fixing the engine part of things. And the side effect of the happy side effect we found is the more training we do, the more business we get out of it. As stated, researchers are busy people. They don't have time to go seek out someone else to do stuff for them. But if students come to them and say, I took a course with these people, they were very helpful. Can you get them to help us with this other thing? That is a great way to get in the door to show what you can do for them. So training has been a fantastic means of advertising. We've had long relationships with several labs that have been years long, some of which are not even located in Ottawa. And that's fantastic because what we can do is we're deeply familiar with their projects. We have all their stuff on hand. So if they need, there's an analysis we've done in the last couple of years for them, and they need new figures done, that can be done in a matter of an hour or two sometimes, simply by pulling up their project and updating it. So having that ongoing relationship is great. It also becomes a piecework thing where they just ask us to do stuff and we build them for the time and build them quarterly. So that as a relationship has been excellent. But it took a long time to develop that kind of relationship with the groups. We work in close collaboration with STEM Core, to a lesser extent with other labs, but STEM Core is a sequencing core of the University of Ottawa and the Ottawa Hospital Research Institute. So when, as I said, we get in at that ideal phase, at the grant application phase, we'll often work with the sequencing facility. We'll go to the scientists, we'll find out what their budget is. We'll work with STEM Core to design an experiment that fits within their budget, that tests their hypothesis. And to be able to do those at the same time is great because it reduces the work on the scientists and it really optimizes the effort on the part of the course because we know how to work with each other. And then the final thing is that I really have been happy about, and is one of the things I like to, it's been my hobby horse, is scientists spend a lot of time generating new data, but there's huge amounts of old data out there in archives. We've done several projects now for labs where they have a model disease of interest or something like that, or a development phase of interest, and we will integrate huge amounts of data for them on that so that they have a reference data set they can pick through. So we'll take raw data, analyze and integrate it in a uniform manner so they can look through that data and sort of notionally test hypotheses in that data set they have to better frame experiments they'll do in the future. So we've had several successful projects in that area. And that's pretty much my presentation. I'd be happy to answer any questions people have.
Thank you, Gareth. Yeah. So if you have questions for Gareth, please either use the raise hand thing in Zoom, or I think you could just on mic and ask, or put them in the chat.
Just
Diane, go ahead.
Hi, I'm just so that was great, and I'm really curious about your training. So we offer a lot of training here at UBC as well in bioinformatics, and we found we have a lot of requests to do very basic training. So, you know, Linux and R and RStudio and what's a database and that type of thing. Do you have a similar experience?
So we have primarily been doing the RNA-seq course, kindly funded by the Stem Cell Network, but that's done in R. And so we ended up having to do R training for that. So we did some introductory to R training, sort of quick tutorial on that. And we've also done another one, which is a pure R course, which is a sort of month-long one that it's once a week. But yeah, this is the biggest difficulty with these things is you'll get, let's say 30 people wanting to do some bioinformatics analysis, and half of them will be biologists with some computer interests, but the other half will be computer people with some biology interest in this incredible range of skills. So yeah. It's something it's difficult to figure out how to deal with. I agree. I think if you look at software carpentry courses, that's a pretty common one that'll show up where people are helping provide basic skills to bootstrap people into the point where they can do something useful. Right. But so does that answer your question? No,
that's great. It's good to know that other people are having similar challenges. Trying to get everyone to a level where they can do the next, because we've done RNA as well, and it was like, I don't know how to do that in R. Like, okay, well, let's take it back. Yeah, thank you.
Welcome.
Okay, let's go to Vidu and then Laurence, and then I think we'll move on to the next presentation.
Thank you.
Go ahead.
Thank you, Gareth. Great presentation and congratulations for your Platform Scientist Award. I wanted to ask like a question because you mentioned that you are also on the hospital side, and I just wanted to learn a little bit of how you have been successful in having collaborations with industry, because I think that's another one of the areas that I have learned that in many hospital sites, it's kind of a bit of a very low priority, but that's also one of the high earning source as well. Like, if you have those clients, then it kind of covers a lot of cost. Like, have you been successful in having some kind of industrial collaborations for your core? Thank you.
Yeah, thank you. So it really depends what you mean by industrial. We've had a few companies we've done stuff for. For the most part, it's been through scientists have been working for the companies and then asked us to do something. We have worked with government, like both federal government or crown corporations, and that has been, as you say, it's more revenue, but those have just fallen on our doorsteps. We haven't really done any pitches or cold calls for them. I haven't had any. If you have any ideas of how to get that sort of business, I'm not sure. Yeah, so hopefully that answers your question.
Yeah, no, I think like you're right. Like, we do get people coming in from, like, if there are industrial collaborations with the researcher led, then that's kind of like an automatic thing that can come over, but not necessarily like if a industry reaches out to you for a project, then I was kind of interested in knowing, like, if you have had those kind of direct intake of projects from the industry. So if not, then that's fine. I get it. Like, I think it's another.
I wish it'd be nice.
Okay, Laurence, please. Last question for Gareth.
Hi, Gareth. Congratulations for the award. I had a question that I think is a concern for a lot of people regarding research data management plan, and how, depending on the technology or depending on the science that you're doing, might be very, very different. So what's your feedback on what people do and how much time it takes? And is it something that comes along smoothly, or have you seen any particular challenge that might be interesting to share with people around here?
I'm not sure the question in terms of time estimation, you mean, or?
It can be like, yeah, because research data management plan can have different aspects. So it could be either because you have a lot of data or you don't have the right data to find them. Do you promote FAIR principles, and how is it going from the lab's perspective to implement that? Because it takes a lot of time or sometimes expertise. So I just want to have what you feel with the customers you have right now, the users. Is it something they do easily, or are they just learning right now? Or
So for most of the data, we're lucky that in bioinformatics, specifically the sequencing technology data, most journals require that people deposit their data. So we have deposited literally thousands of samples in Gene Expression Omnibus with NCBI of all different types of data, microarray, ChIP-seq, RNA-seq, things like that, some proteomics data. I don't think we've ever had problems with people not wanting to deposit their data or make their data public. It's often just a question of where it might be appropriate to put it so that they can make it available. So we'll often support, well, actually, that'll often be part of the grant of the quote is actually, generating the data, having STEM Core generate the data. We'll analyze the data and we'll deposit in Geo on their behalf and then transfer ownership to them. So yeah, we're very lucky. Bioinformatics has been a bit ahead of the curve on that. So for the most part, and scientists we work with are keen on it, because in my experience, data is often one of the biggest sources of citations. If you get a nice data set, people may use the data for something else completely, but cite your paper just because it got a nice data set that they used. Hopefully that answers.
Thank you, Gareth, and thank you for your presentation.